Hello. How are you, everybody? Here I am. እንግዲህ በአዲስ ርዕስ መጥቻለሁ ዛሬ። Today, I'm going to present the virology section of Unit 2, Grade 12, viruses, about viruses, microbiology, of course, the second unit talks about microbiology, and we have to uh, see viruses, the different microbes. We have seen archaea, bacteria, and now we should see the viruses. Therefore, without much ado, let's go uh, to our lecture. Let's start our lecture today with this question. What are viruses? If you are asked this question, how would you respond to? Of course, viruses can be defined as viruses are a group of ultra microscopic, um, non-cellular organic entities, highly infectious agents, and that multiply only inside a living host cell. That is, they are obligate intracellular parasites, you see. Therefore, these are the descriptions of viruses. Non-cellular, ultra microscopic, which means very much microscopic, which can't be seen even by the latest compound light microscope. Unless we use the electron microscope, we can't see them. They are non-cellular, or simply they are organic entities. They are organic molecules, biological organic molecules. Principally, of course, a fully developed infectious form of virus, which is known as virion, is principally composed of protein coats and nucleic acid, either RNA or DNA, but not both. And they are highly infectious agents, of course, and they only multiply in living cells. That is, they are obligate intracellular parasites. Then, are viruses living or non-living? Which would you prefer? We can say living, and we can't also say non-living. They do have both features or characteristics viruses to have. Considered non-living because, you see, they are acellular. In biology, uh, cell is the smallest structural and functional unit of life. Once they are not cellular, we can consider them as organisms. To be organism, at least should be a cell. Therefore, they are acellular, do not have cellular organelles or cellular structures. They can be crystallized. You see, this is a non-living feature of viruses. They can be crystallized as salt crystals or sugar crystals. They can be crystallized as chemicals. They possess only one type of nucleic acid, either RNA or DNA. Of course, all living organisms, which means all cellular organisms, what we call organism, must be made of cell. Depend for gene expression both nucleic acid, DNA and RNA. When it comes to viruses, viruses contain either RNA or DNA, but not both. They only is composed. They can reproduce in living cells only by containing their genetic material in the form of DNA or RNA. The other do not metabolize energy. They don't produce energy, but they use the energy made by their host cells. They don't have the machineries and the enzyme system for energy production by respiration. Can't grow. There is no growth here unless they multiply, of course. They reproduce, multiply, they increase the number. There is no phenomenon known as growth, which is seen in, which is common property of living organisms. They can't reproduce on their own. All are parasites, and they are obligate intracellular parasites. Just obligate intracellular inside host cell parasites. Outside of a host cell, we can consider viruses as simple organic entities which are non-living. On the other hand, viruses belong to biology because they show some living traits or properties, such as They possess genes or nucleic acids and proteins. These are biological molecules. All living organisms contain nucleic acids and proteins. The fact that viruses are composed of biomolecules, nucleic acid and protein, makes them, just gives them biological traits. The other can replicate, they can reproduce, though they need, they must enter into living cells, they can reproduce. They can hijack the host cell resources and machineries, and can reproduce and can evolve. This is one feature that makes them share living features. That means evolution. They can evolve. They can undergo genetic material change in course of time, of course, and they can evolve. All living organisms evolve. There is evolution, slow and gradual change. Yes, they are adapted to particular hosts or biotic habitats. You see, they even they can they change the host cell environment to be suitable for them, which means they can get adapted to the host cell so that they multiply and come out of the cell to repeat the cycle. Therefore, they are adapted to particular hosts or biotic. Adaptation is the feature of living organisms, of course. Um, Outside the host cell, they are inert particles, non-living, and an inert extracellular form of a virus, as I've tried to tell you earlier, known as virion, of course, a fully developed extracellular form of virus, infective form of a virus. You can define virion like this. Thus, viruses are regarded as intermediate between non-living entities and living organisms because of the fact that they possess non-living properties as well as living properties. Viruses are obligate intracellular parasites only able to multiply inside living cells by using the host cells resources. I mean, metabolic machineries. I've tried to tell you earlier this. Um, in other words, they are non-living infectious entities that can be said at best to lead a kind of borrowed life. They are totally dependent on their host cell. They are organic entities which are just like living or leading a kind of borrowed life, borrowed from their host cells. Unless they are inside host cells, they can't reproduce, they can be considered as non-living. Viruses are therefore parasites of cells and are dependent on their hosts for most of their requirements, including building blocks such as amino acids and nucleotides, protein synthesizing machinery like the ribosomes. They use the ribosomes of the host cell. Energy in the form of adenosine triphosphate. They use the energy made by their host cell. They can't metabolize energy. A virus modifies the intracellular environment of its host in order to enhance the efficiency of the replication process. Imagine they are highly effective in this regard. That means once they enter into the host cell, they can change the host cell environment, the cytoplasm, including the nucleus, if they are eukaryotic cells, to suit the environment to be suitable for them, so that they can multiply and reproduce and come out of the cell. Highly specific about what they invade. The other fact about viruses, they are highly specific about what they invade. And for example, plant viruses infect only plant cells. Bacteria viruses, which are also known as bacteriophages or simply phages, infect only bacteria or prokaryotic cells. And human viruses, such as HIV, infect only humans. Let's go to the other topic, structures of a virion. That means virion is a fully developed extracellular form of a virus or an infectious form of a virus, principally composed of what? Two components, capsid, protein coat known as capsid, and core, the inside nucleic acid, either DNA or RNA. Let us see the structures of a virion. Virus outside of a living cell, extracellular form of virus is called virion or viral particle. That's a virion or viral particle. And all virions are composed of two components principally. One is capsid, as I have told you earlier. Capsid is protein coat. And inside, nucleic acid, core, the inner part is the nucleic acid, which is from the inside attached to the protein coat, the inner layer of the protein coat. Therefore, capsid, let's see capsid. Capsid is a protein coat that covers the viral nucleic acid core, yes. And capsids are built from large numbers of protein subunits, usually protein subunits, protein bricks known as capsomers. The units that are making the coat are protein units which are known as capsomers. The capsid may also enclose some viral enzymes together with the nucleic acid. Therefore, if you are asked what are the structural components of a virion, one is the coat. The coat is made of protein, and this protein, the subunits of protein are known as capsomers. These capsomers join together to form the coat in a symmetrical fashion, of course. We shall talk about the symmetry of viruses. Then the capsid may enclose some viral enzymes together with the nucleus. Of course, some viruses may also contain inside the core, together with their nucleic acid, they may contain specific enzymes, the enzymes of the virus, vital enzymes of the virus. The second component is nucleic acid core, the inside. The nucleic acid of a virus is the carrier of its genetic material, and it is either DNA or RNA, but not both. Even the DNA is, it could be double-stranded DNA or single-stranded DNA, which is quite different from the living systems. Even the RNA, also RNA viruses, also are divided into RNA virus, single-stranded RNA and double-stranded RNA virus, of course. We shall talk about classification based on DNA and RNA nucleic acid classification later on. Therefore, the nucleic acid is DNA or RNA, but not both. The DNA, single-stranded or double-stranded. RNA, double-stranded or single-stranded. You can. And the nucleic acid composition of a virion is one major criteria for classification of virus. You see, one criteria. Especially, we will come to this point, the Baltimore's classification scheme. American virologist David Baltimore classified viruses based on their nucleic acid. Therefore, nucleic acid composition is one criteria for classification of viruses. Remember, get here, if a new virus is isolated, one way to determine whether it is an RNA virus or a DNA virus is to test its susceptibility to a ribonuclease and a deoxyribonuclease. You see, ribonuclease is enzyme that can digest RNA molecules. Deoxyribonucleases are enzymes that digest DNA molecules. Therefore, if you expose a newly discovered virus in order to see whether it is RNA or DNA exposed to these enzymes, if its genetic material is digested by ribonucleases, therefore it is RNA virus. If its genetic material is broken down or digested by deoxyribonucleases, it's DNA virus simply. This is simple mechanism to see whether it is RNA or DNA virus. The virus nucleic acid will be susceptible to degradation by only one of these enzymes. Why? Because every virus, any virus, contains either RNA or DNA. So far, no any virus found which contains both the nucleic acids. Okay, And in addition, the genetic material of viruses could also be either single-stranded and double-stranded. I have said it earlier. Thus, there are four possibilities for a virus genome. One, double-stranded DNA. Two, single-stranded DNA. Three, double-stranded RNA. Four, single-stranded RNA. These are generally four major. These are the four major nucleic acid composition of viruses, of course. Envelope. You see, this is optional. Envelope is not common to all viruses. Some viruses, which are known as enveloped viruses, also contain an outer an outer covering, which is phospholipid covering, which is derived from the host cell but modified by the insertion of some glycoproteins of the virus. Envelope. This belongs to only one group of virus, which are known as enveloped viruses. Uh, some viruses, it says, in addition to the above two components, which capsid and nucleic acid are, may contain an outer lipid membrane covered now lipid membrane covering known as envelope. And this envelope is derived from the host cell. I've told you the host cell membrane in which they multiply. When they multiply, at the end, they modify the cell membrane of the host cell and use it as envelope and cover themselves and come out of the host cell. However, the viruses insert their own proteins or glycoproteins known as peplomers or spike proteins. These are glycoproteins, of course, carbohydrate residues. You may call them simply proteins or glycoproteins, peplomers or spike proteins. In some viruses, of course, this could be pure proteins. You have to not forget. And function of this spike, the function of this envelope, the envelope contains spike proteins, peplomers, the spike proteins. This spike protein, the spikes typically have a role in viral attachment. When next time the virus goes to the host cell, the first step, remember, the first step in viral replication is attachment. Therefore, the spikes are designed for attachment in the host cell membrane. The attachment is not a physical attachment. It's a matter of covalent bond formation, a strong bond formation. Thus, viruses that possess envelope are known as enveloped viruses. Viruses without envelope are known as non-enveloped viruses or necked viruses. And you can see here, you can see simply the envelope. You can see the envelope to the left, the necked virus, capsid, capsomers. The spherical bodies are units, protein units known as capsomers. Inside, you can see nucleic acid. There is no envelope. The diagram to the left, or the virus to the left, I mean to the right, has envelope. This envelope has the black beads, which are known as spikes. The spikes are made by the virus. The blue region is the phospholipid bilayer of the host cell, of course, cell membrane. You see, therefore, envelope and envelope proteins determine biological properties of the virus. You see, envelope is lipid, so it is susceptible to the action of detergents. You see, you remember during the time of COVID coronavirus. COVID-19 is sort of coronavirus, which is of course single-stranded RNA virus. It is enveloped. It's enveloped. You see, using soap to wash our hands as hygiene to prevent transmission of this virus during the COVID-19 time, is that the envelope, the outer layer of the virus, can be easily dissolved by the soap, detergent, the soap. So we can remove the coat, the envelope. If the envelope is removed, the virus has lost its spikes in the envelope, which means it can't attach to the host cells inside our in our upper respiratory tract, of course. So unless it attaches, it can't enter its genetic material or itself, and it can't multiply. So unless it multiplies, it can't cause disease. This is one way of fighting, controlling the virus transmission. Okay, DNA viruses. Let us see some examples. Herpes virus, pox viruses, hepadenoviruses, HEPA DNA. You see, hepadenoviruses, RNA viruses, flaviviruses, Toga virus, coronavirus, hepatitis. Of course, hepatitis type A, B, C, D, E types are there. Orthomyxoviruses, paramyxoviruses, rhabdovirus, Bunia virus, filovirus. These are RNA viruses in general. And retroviruses. Retroviruses are special types of RNA viruses, the HIV/AIDS or lentiviruses. Retroviruses are RNA viruses, but way after they enter into their host cell, they change their RNA into DNA, double-stranded DNA. You see, retroviruses are single-stranded RNA viruses. They have special protein, special enzyme, reverse transcriptase. By using RNA, they produce DNA. Therefore, these are enveloped viruses that we have seen, DNA, RNA, retroviruses. Now, non-enveloped viruses, DNA viruses, parvovirus, adenovirus, papovavirus, RNA viruses, picornavirus, hepatitis A virus, and hepatitis E virus. These are non-enveloped. I've told you, hepatitis viruses are A to E, A, B, C, D, E. The A and the E are RNA viruses, but non-enveloped. You can see here. You can see the envelope inside, capside and inner core. Envelope, envelope coat of lipid generally derived from host cell membrane. We have said this. Genetic material could be DNA or RNA. Capsid protein coat encasing the viral genome. The capsid is made of capsomers, protein units. And this is another diagram for you to see the enveloped virus and what the outer layer decorated with glycoproteins or envelope proteins. You can also call these spikes envelope proteins. Even these envelope proteins are also used in classification of the enveloped viruses. Okay. Now look these viruses, bacteriophages. These viruses are specific to multiply in prokaryotic cells like bacteria. They can only multiply living inside bacterial cells. They can't attach and enter their genome into eukaryotic cells. They are not able to enter and multiply in any of the eukaryotic cells. They are designed to destroy. Of course, they destroy, destructive to the. Of course, these bacteriophages are also the most abundant in our planet, in the biosphere, the most abundant viruses in our ecosystem. Size. Viruses are ultra microscopic, as we have stated, very much microscopic, which can't be seen without using the electron microscope, and not visible with the light microscope, and require the electron microscope, of course. Range in size 20 nanometer to 300 nanometer in diameter viruses. And this makes them several hundred times smaller than an average bacteria and several thousand times smaller than most human cells, you see, in terms of size. They are very much. The smallest known virus is 20 nanometer in diameter. Of course, recently, here, the the largest the largest virus is given 300 nanometer, but a recently discovered virus, there is a recently discovered virus which is known as Mimi virus, which is around 600 nanometers in diameter so far. Therefore, this is new information which is not included in the literature we expect to be written in the future. You can see comparison of size. You can see only the portion of eukaryotic cell like human cell. Then you can see bacteria. Then up you can see the virus, how much there is big size difference. That's why when one virus enters into human host cell, like HIV, one HIV virus enters into the CD4 cells, multiplies, becomes 100 to 200 viral particles. When they come out, the cell membrane is enough for all these viral particles to use it as envelope when they come out, destroying it using the envelope. After modifying the envelope by inserting spikes, they use it as envelope and come out. This is the end of this session. ቴንክ ዩ ቬሪ ማች። እንግዲህ ቀጣይ በሚቀጥለው ፖርሽን የቫይረስ ፖርሽን እስክንገናኝ ደህና ሁኑ። እንግዲህ ይህን ቻናል ሰብስክራይብ፣ ላይክ፣ ሼር ማድረጋችሁን ፕሊዝ ቀጥሉ። ፕሊዝ ኢንጆይ ማይ ቪዲዮስ። I will come by the next portion next time. Bye bye.